All right, now that we've finished talking about antigens, it is logical to talk about antibodies. So welcome to Unit 6. So our third line of defense is the production of antibodies, or also known as immunoglobulins. These are glycoproteins or gamma globulin proteins synthesized by our plasma cells or B cells. They can be found in tears, saliva, blood, or plasma, and they are used by the immune system to neutralize viruses and bacteria. So an antibody is a protein found in body fluids. They combine with an antigen in order to neutralize bacterial toxins or viruses. Generally, antibodies are all similar in nature except for a small region at the tip of the protein, which is extremely variable. Each of these variants can bind to a different antigen. Antibodies contribute to immunity in three ways. They prevent pathogens from invading by binding to them. They stimulate macrophages and stimulate the complement pathway. B cells become either antibody-producing plasma cells or they become memory cells. A memory cell is a long-lived lymphocyte capable of responding to an antigen on its reintroduction long after the initial exposure that caused its production. Now, antibodies can be found in mucous membranes, serum, saliva and tears, breast milk, and spinal fluid. And there are 5 classes of isotypes or antibodies recognized in higher mammals. The major serum antibody is IgG, but we have IgM, IgG, IgA, IgD, and IgE. They are named after the heavy chain from Greek words such as mu, gamma, alpha, delta, and epsilon. These antibodies differ from each other in characteristics like molecular weight and carbohydrate contents. The basic immunoglobulin molecule is bifunctional, so one region binds to an antigen and a different region of the molecule mediates binding of the antibody to the host tissue. So each arm can bind to a specific antigen, and they are divided into 2 groups, the antigen binding region and the antigen that binds to the host tissue and other host structures. A good example of a typical immunoglobulin molecule is the immunoglobulin G molecule. Okay, so let's take a look at the immunoglobulin G molecule up here on the slide. Okay, the primary core of an antibody consists of the sequence of amino acid residues linked by a peptide bond. All antibodies have a common basic polypeptide structure with a 3-dimensional configuration. Antibodies are characterized in class according to the heavy chain. Okay, the heavy chain consists of 446 amino acids, while the light chain only consists of about 214 amino acids. All amino acids are bound together by peptide bonds. Okay, you can see on this molecule here that we have the 2 heavy chains and the 2 light chains. The heavy chains are either gamma, mu, alpha, delta, or epsilon, and the light chains are either kappa or lambda light chains. Okay, in any monomer or any molecule, usually you will have 2 kappa or 2 lambda light chains. You will not have 1 of each. You will have 2 of each. Just a little more when it comes to the anatomy of an antibody. We've got the Fab region, which stands for fragments antigen binding. It's the molecule end that binds to an antigen and recognizes specific foreign objects. We have the FC region, which is the fragment crystallizable region. This part will actually crystallize when purified, and this segment contains only heavy chains. This portion binds serum complement, contains properties that dictate if the molecule can cross the placenta, and it directs the biological activity of the immunoglobulin. So the bottom half of the heavy chain is called the FC region. And it modulates immune cell activity, binds to specific cellular or tissue proteins, mediates different physiological effects like cell lysis and degranulation of cells. Then we have the FD region, which stands for fragment determination or fragment determinant. This is part of the heavy chain component of the Fab region that determines the antibody specificity and carries certain well-defined genetic factors. The fragment determinant region, or the FD region, is the top part of the heavy chain only, and it determines immunoglobulin specificity. So when looking at an immunoglobulin molecule, we can look at two different components of that molecule, and we have the heavy chains and the light chains. There are 5 classes of heavy chains, and there are 446 amino acids in the entire heavy chain. The first amino acid through the 114th amino acid are variable, and then 115 through 446 are constant. The heavy chains are called IgG, which is gamma, IgA, which is alpha, IgM, which is mu, IgD, which is delta, and IgE, which is epsilon. There are some heavy chain diseases, and they are generally associated with B-cell neoplasms or cancers, B-cell cancers. We have light chains that are either going to be 2 kappa or 2 lambda light chains, and there are 214 amino acids in the light chain. 1 through 107 are variable, and 108 through 214 are constant. There are different types of light chain diseases, including light chain deposition disease. They might make too many light chains that can be deposited in the kidney. Or they can have kappa or lambda light chain diseases. Some have multiple myeloma. Others develop a non-specific monoclonal gammopathy. Let's go ahead and talk about the 5 different types of immunoglobulin molecules. And let's go ahead and start with IgG. IgG is the most abundant serum antibody. It is usually in a concentration of about 800 to 1800 milligrams per deciliter. This is the major immunoglobulin found in serum. Okay, it constitutes 75% of circulating immunoglobulins in serum. It consists of 2 light chains and 2 heavy chains, and it appears in serum about 7 days after exposure to an antigen. It's the second class of immunoglobulin to form immunity, and it can cross the placenta. It's the only antibody that can cross the placenta from mom to fetus. Without the IgG molecule they get from mom, newborns would have little or no immune protection. Adult IgG levels usually aren't reached until the age of 16 years old. Some in vivo activities of IgG is that it activates complement, enhances phagocytosis. Remember, IgG is a potent opsonin. Okay, an opsonin is something that alters the surface of an organism to be phagocytized. Okay, IgG is antibacterial and antiviral and antitoxin. In vitro activities of IgG would be agglutination. It doesn't agglutinate as much as it precipitates and hemolyzes in serological analysis. There are 4 subclasses of IgG, including IgG 1, 2, 3, and 4. And you will see increased levels of IgG in diseases like hepatitis, rubella, infectious mononucleosis, rheumatoid arthritis, systemic lupus erythematosus, and Hodgkin's disease. Okay, you can find IgG in serum, cord blood, and spinal fluid. The IgM molecule represents about 5 to 10% of globulins and accounts for 10% of the immunoglobulin pool. This pentamer consists of 10 light chains and 10 heavy chains. It's the largest of the immunoglobulin molecules in size, and it has 10 combining sites with many atoms of hydrogen, so it's polyvalent. There are 5 monomers held together by a disulfide bond. It is the first antibody detected after a primary immune response, and usually that antibody is detected about 4 to 5 days after they've been exposed to an antigen. Okay, it's the first antibody class produced by maturing B cells, and it will not cross the placenta. It is most often formed in response to stimulus by gram-negative bacteria. Now, the normal adult levels of IgM are about 60 to 280 milligrams per deciliter in the serum. So there's not much observed in the tissue or other body fluids because of their large size, and it is undetectable in spinal fluid unless the spinal fluid protein is elevated. Okay, in vivo activities of IgM would be antibacterial and will activate complement, enhance phagocytosis, and macrophage activity. The in vitro activities would be agglutination and hemagglutination. In the lab or in serology, it is easily observable due to its large diameter and multiple combining sites of pentamer structure. IgM is elevated in cases of bacterial diseases, infectious mononucleosis, leprosy, malaria, and collagen disorders like scleroderma. IgA represents about 15 to 20% of the globulins. It is found in body secretions, including serum, but the serum concentrations are low. It is primarily found in saliva, tears, the bronchial area, GI tract, sweat. It is synthesized in plasma cells of the respiratory tract and intestine and excretory glands. It is the second most abundant serum antibody, and it is found in body secretions like I already covered, and it protects mucosal surfaces from bacterial invasion. Serum IgA is a monomer, and secretory IgA is two molecules held together by a J chain. This is a polypeptide that allows IgA and IgM to be secreted in the mucosa. Secretory IgA is a dimer. Okay, it will not cross the placenta or activate complement, and most is secreted in the mucosal surfaces. This is found in normal levels at about 140 to 420 milligrams per deciliter. Serum IgA is antiviral and prevents bacteria from adherence to body surfaces. Secretory IgA is found in other secretions of the body, is active in mucus secretions, and will block bacterial adherence and has antiviral properties also. IgA antibodies neither agglutinate particles in vitro nor activate complement in vivo. You can see IgA antibodies increased in things like tuberculosis, rheumatoid arthritis, and liver disease. The secretory component is not bound to any protein in serum. The S component is derived from epithelial cells, not from the plasma cell that has synthesized IgA. The secretory component protects IgA antibody for degradation of gastric acids and enzymes in the mucosa of the GI tract. IgA couples with the secretory component just prior to secretion of IgA into the lumen, and this protects the molecule from digestion from the GI enzymes. IgA is synthesized by plasma cells in the mucosa-associated lymphoid tissue. As it passes through epithelial cells, it picks up that secretory component. IgD is the least understood of all the antibodies. There are only minute amounts in serum, less than 1%. Most IgD is located on the surface of unstimulated B cells and may actually aid in their maturation. IgD does not cross the placenta. It does not fix complement. It is a monomer that consists of two heavy chains and two light chains, and it may play a role in B cell development. It could actually signal the B cell to activate and may bind to basophils and mast cells also to activate these cells in the respiratory disease defense. IgE is the last immunoglobulin that I want to talk about today, and there are minute amounts in serum at less than or about 1%. Almost all of the IgE antibody is attached to tissue mast cells, and some are present on basophils. A mast cell is a tissue cell that contains histamine and heparin and plays a role in allergies. It is elevated in serum from allergy patients, and it's also elevated in parasitic infections. It is responsible for allergic reactions. It does not cross the placenta, does not fix complement, and is monovalent, meaning it only has one combining site.